Okay, we might start now. And Reki, if you can help with managing participants arriving in the chat, that would be great. But since I think we have a pretty packed schedule, we might start right away because we have a lot we want to hear from Koustubh and David. So welcome everyone to our third Snow Leopard Network webinar this year. After hearing updates from China and Mongolia, now we're moving to more of a global perspective on snow leopard conservation. And we believe it is important to keep these two perspectives, the national and the global, in focus because they interact and they help us work towards shared approaches for snow leopard conservation based on real country experiences. And as a network, we're really trying to encourage shared methods and approaches so efforts can really scale up. So today we have the pleasure of inviting David Borchers and Koustubh Sharma, who have been instrumental in formulating the GSLEP PAWS effort. I'm sure many of you guys are aware what PAWS stands for. It's the Population Assessment of the World's Snow Leopards. So David Borchers brings us a wealth of experience in developing mathematical approaches for ecology. He is a professor at St. Andrews University in Scotland, and his team are at the forefront of spatial capture-recapture methods. We also have Koustubh Sharma. He's the GSLEP coordinator and has seen the development of PAWS from its very beginning as a concept to now it's actively working with country teams across the range, turning it into a reality. Koustubh is also supporting the building of bridges between snow leopard conservationists and all snow leopard range governments in bringing snow leopards to the policy level, which is, I think, a fundamental step for the future of snow leopards across the range. So before we start, we would like to ask everyone to keep your mics muted and feel free to write any ideas, any comments in the chat section. So Reki is there to support the Snow Leopard Network coordinator, and she's there if you need anything and anything with issues with the sound, please just let us know. And we expect, you never know, but we expect that speakers will, Koustubh and David will talk 15 minutes each, and then we'll move into a discussion section and see how it goes. I have to alert you though, David and Koustubh, and I think I already told you, I might also have a few questions because PAWS also affects a lot of my work, and it's hard to get hold of both of you in the same room. So I really am happy that you both have come today so that we can ask you questions and discuss this effort. So without further ado, I think first we're going to Koustubh for his first presentation. So go for it, Koustubh.
Thank you, Justine. Just 1 second. Can you guys see my screen okay?
Perfect.
Yeah.
Excellent. Great. Good afternoon, everyone, and thank you, Snow Leopard Network, especially Justine and Raki, for giving us this opportunity to talk about this exciting initiative being coordinated under the Global Snow Leopard and Ecosystem Protection Program. For me, it's an added special privilege to share this this discussion with Professor David Borchers, who happens to be the director of the Center for Research into Ecological and Environmental Modeling at University of St. Andrews. And in addition to that, he is one of the finest teachers you can ever come across. In the next 30 minutes or so, I'll be talking a little bit about why PAWS came into being, why is it following a certain workflow, and why are we developing tools and training workshops around it. David, on the other hand, will take us through an intricate journey to the world, into the world of spatial population ecology. So let's start with the why of PAWS. Now, whether it is the prime minister of a country or a taxi driver, the first question they ask you the moment you reveal yourself as someone working on snow leopard conservation is, so how many snow leopards are there? Despite more than 30 years of solid research and some of the most successful groundbreaking community-based conservation programs, we still do not know how many snow leopards are there in the world. The numbers vary from one educated guess to another, and you can imagine the plight of a decision maker to have to rely on guesstimates when it comes to making policies or taking action. So in 2017, the heads of states or their representatives from each of the 12 snow leopard range country came together in Bishkek for the International Snow Leopard and Ecosystem Conservation Forum. They endorsed what we know as the Bishkek Declaration 2017, which listed the need for a scientifically robust estimate of the global snow leopard population. On the instruction of the GSLEP steering committee, which is represented by environment ministers from the 12 snow leopard range countries, we developed a draft strategy to implement PAWS. Essentially, the task in our hands was to estimate how many snow leopards are there in these 2 million square kilometers spread across 12 countries. Now, if we had the money and human resource, a back of the envelope calculation estimates that we needed nearly 30 million US dollars to cover an entire range with camera traps. And even then, we can't be sure how many snow leopards will decide to not get their selfies taken on our cameras. The good news is that we don't really need to, because this is where statistics can come to our rescue, or at least so did we think. Most of you might know this famous curve that denotes the Dunning-Kruger effect for innovation. Now, if you can plot wisdom from being ignorant to a guru on the x-axis and confidence on the y-axis, this curve provides a fascinating path the human mind goes through when pursuing a certain goal. Now, let me explain this with the analogy of flying a drone, which is something that I love doing. Now, when you don't know about drones, you obviously have no idea what drones are and what it would take to fly it. So you're basically ignorant and have minimal confidence about flying drones. Once you see someone fly a drone, read up the manual, you suddenly feel that you have all it takes to fly one, and your confidence shoots up rapidly. Unfortunately, this leads you to the peak of Mount Stupid, because this is where you realize that it's no child's play and you crash your drone, breaking it badly, and your confidence dips to the lowest ebb, the valley of despair. Having gone through the valley of despair is when you slowly start on the slope of enlightenment. You train, you practice, you partner, you collaborate, and ultimately reach a plateau of sustainability where you hope to slowly grow in confidence, but at a much reasonable and a pragmatic pace. Now, snow leopard is an elusive species that is difficult to be seen. Plus, they have massive home ranges that make it virtually impossible to follow them, let alone count them. To save ourselves from the misery of going through the peak of Mount Stupid and Valley of Despair, we collaborated right from the beginning, bringing experts to fly us from the starting point to the slope of enlightenment as soon as possible. This is why, on the approval of the steering committee of the GSLEP program, we empaneled 12 experts from the field of snow leopard ecology and statistical ecology. And who better to lead this panel than the daddy of spatial capture-recapture, David Borchers. David and his esteemed panel, including others such as Luja, James Nichols, Charu Mishra, Daryl McKenzie, Sandro Lovari, Chris Sutherland, Somali, Tom McCarthy, Wali Mudakik, and Ashik Ahmed Khan, came together and developed a detailed roadmap. The panel gave us a much better clarity on how to achieve the Herculean task and what to expect versus what not to expect. What became exceedingly valuable to us was to notice that PAWS was not going to be just about snow leopard populations. Done the right way, it could lead us to achieving several additional goals. For instance, we can now end up with a refined, accurate distribution map of snow leopards, something that I will talk briefly about in a few minutes. Moreover, it can help us map threats, build capacity for young conservationists and frontline staff, identify climate refugia, and estimate prey populations in several regions. All of this, in addition to methodological developments in population ecology, improving our understanding of snow leopards, population dynamics in general, and also to know the efficacy of conservation programs. The very first step to implement PAWS requires planning. Planning at a regional or national scale. Now, why? Because we need to identify who can and will do what, when, and how. This stage essentially brings together partner organizations and governments to create a collaborative framework where necessary. It is delightful to note that each country is using different means to develop implementation modalities under PAWS. Very quickly into the PAWS process, we realized that to know how many snow leopards there are, we need to clearly know where they are. One may ask, why do we need this when we already have snow leopard distribution maps prepared by experts, or when there are several models using presence-only data? Unfortunately, none of the existing maps take into consideration the fact that snow leopards can at times be in a particular area and yet not be seen. In other words, absence of evidence is not always an evidence of absence. The very recent surveys across Mongolia that Gantulga and Puji talked about last week revealed that both the expert opinion-based as well as presence-only data-based snow leopard distribution maps that we have are inadequate. Snow leopards occupy areas that we didn't earlier know about, and conversely, they're also missing from areas we earlier assumed them to be present in. One of the primary aspects of PAWS is to count snow leopards. Silly question. Whether you use genetics or camera trapping, the best methods, as you will see shortly in David's presentation, require us to identify individuals. But then someone may say that we already have more than 24 sites from where we have peer-reviewed studies reporting snow leopard abundance. Why can't we simply extrapolate that number to estimate their global populations? Again, let me take you back to Mongolia one more time for a minute. Mongolia is not only the country with the second largest snow leopard population, it is also a country that exemplifies strong collaboration and partnership between NGOs, institutions, and government. The black patches you see here represent about 26 camera trapping studies that have already been conducted in Mongolia in the past couple of years by WWF, Snow Leopard Conservation Foundation, Wildlife Conservation Society, Protected Area Management, independent researchers, and so on. Cumulatively, these represent more than 100,000 square kilometers, which is nearly 25% of the entire snow leopard range or entire projected snow leopard range in the country. Now, in a normal scenario, one would assume that with such a coverage, the data should be sufficient to get reasonable estimate of snow leopard abundance. Just for comparison, look through the lens where we know snow leopard abundance estimates from just 1 or 2% from across the snow leopard range. Now, this is where a stats guru used the field data and developed a hypothetical surface showing expected number of snow leopards per 15 by 15 kilometer grid cell that Gantulga showed last month. They assumed that a mean density of just about 1 snow leopard per 200 square kilometers, really half of what we know to be the density in many parts of the range. The first simulation produced a snow leopard population of about 1,300 individuals. Using a simulated camera trap study of the population, they estimated the snow leopard density for each grid cell. This estimated density was then used to estimate abundance across the entire country by multiplying the total number of grid cells that are likely to be occupied by snow leopards. What we found was that the net effect of using only the existing camera trapping data as if these were representative of the whole region can lead to substantial bias. The estimate of the size of the simulated population in the map that you see here, with a known abundance of about 1,300 individuals, was more than 1,600 individuals, a clear overestimate of 25%. We ran the same simulation 500 times, each time using spatial capture-recapture analysis to estimate abundance. The histogram on the right shows that the range of percent errors on an average still recorded a mean positive bias of 23%. Now, it is because the current trapping effort is inadvertently sampling only the most suitable snow leopard habitats. On the contrary, if you use a balanced sampling effort then you generate a mean error of nearly 0%. Now, what does that mean? Do we deem existing studies redundant? Do we start all over again? Is this the value of despair? No, you just balance your future samples, that's all. But still, you get to use the existing sampling. How do you do that? This is where David and Ian, who was here last month, have developed this very clever tool to develop a sampling approach to estimate snow leopard abundance from large, massive landscapes. Feed in your existing knowledge, data, and availability of resources, and it will pop up guidance on where to sample next. So this was just a glimpse of one of the numerous other tools, training programs, and applications that are gradually being developed and rolled out on the GSLIP website's PAWS section. Why? Because as a collaborative initiative, PAWS needs the support and collaboration of a multitude of teams working on the field, in the lab, and on their desks. With that, I think I've kept my time. Yes, I'd like to pass it over to David, who can take you now to the intricacies of spatial capture-recapture process that is truly at the center of the entire PAWS process. Thank you.
Okay, just trying to share my screen. Just bear with me for a second.
Thanks, Koustubh, for the, and I see some questions coming up in the chat. Please feel free to start the questions and we'll answer them after David's presentation.
Okay, so that's my screen share. Can I just check that it's the right screen? It's a presentation, not the other one. Yeah.
Yes.
Okay, so what I'm going to be talking about is sort of the basics of spatial capture-recapture. And I'm going to do that without any equations at all. And I'm going to do it with using this example of a survey in TOST. So the picture that you now see is the TOST survey region. The red dots are camera traps, there are 40-something of them. And the colors are a measure of ruggedness. So the yellow areas are the highly rugged, so that's up in the mountains, and the blue areas are less rugged. And that's something that we expect a priori density to be related to, and possibly also animal movement to be related to. So from these cameras, you get data that, I'll talk about the data in a second. I'll talk briefly about what the spatial capture-recapture models are like. There are 2 bits to a spatial capture-recapture model. There's a density model, so that's the model for where the animal's activity centers are. And thinking back to that picture, we'd expect more activity centers to be in high ruggedness than low ruggedness areas a priori. And then the second part of a model is the encounter model. So this says, what is the expected number of encounters or the expected detections as a function of how far the camera is from an animal's activity center. So there's these 2 components to the model, one being how do the animals move about essentially, which is the encounter model. How likely are we to detect them at distances from their activity center? And the other one, the density model, where the activity center is likely to be. The kinds of data we get out of spatial capture-recapture come in 3 kinds. You've got to know where your detectors were. So the first kind is the detectors, which I'm going to use the example of camera traps. So they're cameras in this case. And the basic thing you have to say is where they are. You have to give the coordinates. You can then also attach various covariates, so explanatory variables to them. You may have different kinds of cameras. They may be in different habitat. They may have operated for different times and so on. The second and crucial bit of information is the encounter histories or the capture histories. So this tells you the basic you've got to have here is how many times each animal was encountered on each detector. And if you have more than one sampling occasion, then you've got to know this by occasion. You can also have covariates, so explanatory variables at this level. So they'd be attached to the animal, things like animal sex or age, if you can see age, and so on. And then the third kind of data you've got to pass into the estimators is some definition of your survey region. So the boundaries of the region that you were using the cameras to sample within, and some definition of its location. And this is absolutely crucial. In fact, this survey region is something that you really have to define before the study. Quite often people put cameras out without defining the survey region. That really restricts what inference you can make. You must define the survey region first and then put your cameras out to sample from it, according to some of the designs that Koustubh spoke about. Now, within the region, you can also have covariates. So these are spatial covariates, ruggedness in our case, but it could be distance from water or various other things. Okay, so what data do you get out of this? Well, this is a quick summary of the data that was got out of this survey and tossed. These black dots are encounters. You'll see they attach to the traps, which are the red dots, and the height of the line on which the pin on which the black dot is the number of times an animal was captured. So the height of this one, the highest one, is 6. An animal was caught there 6 times. And let me just show you for a couple of individual animals what these look like. So this is animal number 6, who was caught 6 times on this camera over here, once on these two that are dark blue, twice on the one that's light blue, and 3 times on the one that's green. So that's the information we get out of animal number 6. Now you can tell immediately just by looking at that animal number 6's activity center is somewhere in the vicinity of these pins. Animal number 9, their activity center is somewhere in the vicinity of these pins. So these spatial captures tell you immediately about something about where the animal lives. And they also tell you something about how far the animal ranges. So the fact that these pins are all within, I don't know, some kilometer or a few kilometers of each other tells us that the animal doesn't range very far. So we can formalize that by estimating an encounter rate function and how this depends on distance from activity center. And to explain that, I'm just going to go from 2 dimensions here, easting and northing, into 1 dimension just to make it simpler. So this is some hypothetical set of 5 cameras. Camera number 5 got no captures of this animal. Camera number 3 saw the animal 5 times. Number 1 saw it 8 times, and so on. So this tells us something about where the animal lives. Intuitively, we'd say it must be somewhere between 3 and 1 because it got most captures on 1, so it probably lives closest to 1. It also tells us something about how far it ranges because it got no captures on 5, so it's unlikely to range much further than sort of the middle of these captures and the distance from there to 5. And so to formalize that, we just fit a smooth function to this lot. So I've done that here, the bars again in red, and I've just fitted this half normal encounter rate function, and I've estimated the activity center. So the normal shape that fits these red lines best is one that has an activity center at this black dot, and it has a sigma, so a range that makes the curve fall down in the way shown by this black curve to the left and right. So as you move further and further away from the activity center. So that's in 1 dimension, but really what's going on is we're fitting this all in 2 dimensions. So let me just go back to TOST and show you what it looks like for animal number 9 in 2 dimensions. So here we've got this normal shape. It's now a 2-dimensional normal bump, or it's a hill now instead of just a curve. And the black lines here again are the number of times that this animal number 9 was caught. And the curve here is the best fitting normal bump to those capture frequencies of this animal. Now note that even though there's some traps well within this animal's range, not all of them captured the animal. So there's some here right near the middle where the animal is estimated to live that caught the animal no times. And that's important for something that comes up soon. So once we actually don't fit to individual animals, we fit these bumps to all the animals at once. And so if I show you a picture of that, there were 14 animals caught in this survey. And so this is a bump for each of the 14 animals. And the black lines are the same encounter frequencies that you saw when I first showed you these data. So that now we've estimated where these animals, each of these animals live and how far they range. But if we said 14 was the number of animals here, that would be probably wrong because there's a fairly high density of traps in this region where the animals are caught. There's a very low density of traps up here in the northeast and a low density off to the west, only 2 traps over here. So quite likely there's animals that live out here that just weren't caught on these few traps. So now I'm going to use, having estimated this encounter function model, these normal bumps, I now use the density model to predict, well, if this is the density of animals in this region and under a simple model where there's equal density everywhere, I would expect a similar density of animals out to here. So maybe I would expect that there were these animals as well that were missed. And actually I've just arbitrarily filled in 20 animals here. And it's actually quite unlikely that there were as many as 20 animals missed on this survey. But what the statistical methods do is they basically search across, well, what if there were 20 animals? What if there were 10, 5, 8, 4? Search across all possible numbers of animals. And the method attaches a probability to there being that many animals, given that we caught nothing on these two traps and these two traps. And it comes up with the most likely number of animals that there were in the whole survey region. And in our simplest model, we'd assume that density is the same across this whole survey region. But in fact, we don't expect density to be the same. So we can put a covariate into our density model, the covariate being the ruggedness. And if we put a covariate into our density model and then predict density, we get a picture that looks like this. So this is actually log density. So it's a log number of animals per 100 square kilometers. And you can see that we predicted much higher densities where there was higher ruggedness, and low densities, this blue is low density, where there was low ruggedness. The other thing we can do is we can put spatial covariates, so the ruggedness, for example, into the encounter model. So now instead of fitting an encounter model that is a nice, smooth, normal bump like that, in fact, this animal is more likely to move left and right in this picture because that's the region of high ruggedness that it likes, than it is to move north and south because then it has to go into low ruggedness. So we can make its movement dependent on a covariate like ruggedness. And if we do that and fit a model, then instead of those symmetric bumps, we get this kind of thing. So this is for an animal who lives out way to the west. And there's very little rugged habitat here, so it tends not to stray very far from its home, because if it goes far from its home, it's into habitat it doesn't like. Whereas this is an animal that lives way out in the east, and you'll remember that this whole area is covered with high ruggedness. So this animal is quite happy ranging much further because it can do that across high ruggedness, which is sort of terrain that it likes moving through. So that's all just describing a spatial capture-recapture model fitted to a closed population. So we've just been interested in how many animals there are and where are they, so their density and how that varies in space. I'm not going to go into the details at all, but we can do all of that with open population models. So this is not snow leopards now. This is a camera trap study of jaguars in Belize, and there were 12 consecutive years of surveying. And the red curve shows the estimate that we would get were we to fit a closed population model to each individual year. And the blue curves shows the same estimates of the trajectory of the same population were we to fit a population dynamics model. So by that, I mean, we put in demographic parameters. We put in parameters for recruitment and survival effectively. And you can see that we get much the same estimates, but by using all 12 years and putting in demographic parameters, we get a much smoother curve and substantially lower variation. So the grayish blue line here is the variance about the population dynamics model, whereas the reddish stuff is the variance about the series of closed population models. And that pretty much is all I want to say about how spatial capture-recapture works. So the quick summary is that we use those encounters in space to model both animal space use via the encounter model, which could either be the symmetric normal bump or something more complicated, and the animal density, which the default would be uniform, but in most situations, animal density isn't uniform in space. And so we can then also draw inferences about how both the space use and the animal density depends on explanatory variables. And these variables could be at the animal level, so they could vary by sex. Spatial variables, which is the example I gave, so that's varying by ruggedness in the example in TOST. And also in time, which is the example from jaguars in Belize, how density changes across time. And then last comment is that I've only spoken about camera traps here, but we can also do all of this with these spatial capture-recapture methods with genetic samples from scat samples. The only real difference in terms of methodology there is that when you do scat samples, the samplers aren't these points, camera traps, they tend to be lines. You tend to walk along transects. And that's all I want to say about this. I do, if there's time, I can say a little bit more later about some developing areas in spatial capture-recapture and the kinds of things that, additional stuff that we can draw inference about beyond the sort of basic stuff that I've spoken about here.
Thank you, David. No, that was great. And I think we might ask you to go into those after a few questions and a few discussions. So we might take you up on that offer. But before that, as people start writing in the chat, I just wanted to ask Koustubh if you wanted to comment on David's presentation. And maybe, I don't know, you've worked in TOST. So these activity ranges of these different individuals, they look very similar, like animals have been collared and tossed, right? So I just wanted to ask you if you could comment on how this modeled approach using camera trap is similar to the collaring home ranges, if you had some thoughts to share with everyone. And then we'll go to questions from the audience that are coming in.
Yeah, thanks, Justine. I mean, as I said, you know, it's really magical when you see a model behave like what you would expect it to behave. And one modeler, I don't remember who, once said that all models are wrong, only some models are useful. I think that's a famous saying, David, if I'm not mistaken. But to me, I think one of the initial exercises that David and I, when we were working with some of the data, was when one of the models that David developed, it came up suggesting that from point A to point B, a snow leopard is likely to take this path. And when I overlaid with telemetry data, that's exactly the path that it took. Now, model that came up with that information did not have any telemetry data. It was using camera trapping data. So what was fascinating to me in many ways was that just based on some understanding and some models developed with camera traps, we were able to predict animal movement. For example, I'm not even talking about abundances, but just animal movement through a path which exactly overlaid with what path snow leopards were taking based on our telemetry-based studies. So I think that to me was the light bulb moment. I was like, wow, this works. You know, you see the match between field data versus the models that are coming up. So I think that was a spectacular start that we had. We've had many challenges after that, and we will not get into that because, as David said, it's a fast developing and growing field, and we're still, I think, new things are developing as we speak. So, yeah.
So thanks, Koustubh. So we're getting a lot of questions about the type of data being used for spatial capture-recapture. There's a few questions related to genetics and what is the progress in terms of genetics and using for snow leopards in spatial capture-recapture so far. I think it's from Chloe. What has been the success in individual ID from feces to do this kind of work? So I think this question may be first to Koustubh.
Sure. Yeah. So Justine, I'm not a geneticist, so I wouldn't be the right person to really answer this. But there are publications on snow leopards which are using genetic data from feces to estimate abundance. I think there was a paper by Madhu Chetri in Nepal, a pretty large landscape that he had surveyed. Having said that, there are different papers again which claim the difficulties or the challenges in comparing individual IDs using microsatellites that people use. Again, not being an expert, just one little point that I wanted to highlight here, which was there in the last slide that I showed, is that we're also parallelly working with the Stanford University, the National Center for Biological Sciences, and at least 15 other organizations and institutions across the world to develop the snow leopard genome. The hope there is that once we have a snow leopard genome, or what they call as SNIP, single nucleotide polymorphism panel, it is anticipated that our ability to identify individuals will improve multifold. The cost will come down dramatically from nearly $100 to about $8. And it will be possible to do those IDs in relatively less equipped laboratories across the range. One doesn't really need to go to very specialized labs as well. That's what I've understood so far. But yes, so far it has been useful to some extent, definitely. But there's more development coming in. So yep, watch this space, as they say.
Great. And there's another question related to individual ID that builds on that, even related to camera traps. So I want to maybe, David, you can take this question. What happens, you know, they say, if you put trash in models, it comes out as trash. And with snow leopards, their fur is so thick and you get sometimes terrible photos. So the ID process can be a big bottleneck. Do you have any concerns about that? And how and where it is, if SCR is moving towards maybe accounting for such errors?
Yeah, that is, I mean, that's a really important issue, and it's an issue that's really difficult to deal with. The easy way of dealing with it is when at the point you're identifying, only identify stuff that you're 100% sure of. And the only cost of that is that you reduce the sample size. But that's a safe thing to do. The difficult thing is when you include stuff that, an animal ID that you're not 100% sure of, that might be a new animal, or you might confuse one animal with another, then that potentially causes huge problems. There are methods being developed, spatial capture-recapture methods being developed to deal with that. And so I expect in the next 5 years or so, there will be methods that can be rolled out to deal with that. At the moment, the Bayesians are kind of ahead of the game compared to maximum likelihood frequentist people. Just because the way the Bayesians deal with it is you can sort of sample across all possible combinations that there could have been. Short answer, yes, important area of hot research, but quite difficult to solve.
Thanks, David. And Koustubh, I don't know if you want to add on this, on how PAWS is trying to address this, because if we are trying to get accurate estimates, then this is a subject for PAWS. So where is PAWS going in terms of addressing this live?
Yeah, so as both David mentioned and you indicated, Justine, one of the key developments at this point in time is to find a means to minimize your errors. And if I remember correctly, there's a little guidance which is coming out soon. In fact, you should all follow Justine for that, is to try and figure out what needs to be done to minimize errors on individual IDs. So starting with that, there is also parallel development happening in potentially using AI, artificial intelligence, to come up with some sort of a probabilistic identification system. But these are a little further down the line. And as David says, that it may take a few years to get the statistical tools to deal with such data. Similarly, it might take a few years to get to an AI-based solution for individual IDs. But yes, there are certain workflows which you should be able to find on the PAWS resources on the GSLEP website as well, where you should be able to at least find the right way to minimize your possible errors. I think that's what we can aim for at this point in time, given where we stand.
Thanks, Koustubh. And another thought that comes into errors that I want to ask you, David, and this is one of my questions. Sorry, I'm just going to leap in there and ask, related to applying spatial capture-recapture to the snow leopard range. What if we have huge gaps in terms of area of snow leopard range that we do miss out? For example, I've heard estimates that we need at least 30% of the range monitored, for example, and we've only have around 2% at this time, which is what people say. So I wanted to hear your thoughts, David, in terms of applying the SCR versus occupancy PAWS method to the snow leopard range. How do we deal with this big challenge?
Yeah, so you can deal with huge areas that are left out. I mean, it's quite common in many surveys to cover only a very small fraction of the area. For example, before I got involved in things like snow leopards, I was working on Antarctic surveys of whales. And the Antarctic, over a decade or more, managed to cover maybe 2% or 3% of the range of the whales. And we got, for the more abundant whale species, you get reasonable estimates of abundance. Similarly for snow leopards, there's no, without massive resources, there's no prospect of covering a very large fraction of their range. And so the key there is a randomized design. So that where you look is important, and the key thing about where you look is it must be representative in some sense. And the robust way to be representative is representative in the sense that where you looked, you could have looked anywhere. You were equally likely to look anywhere. So therefore, by chance, you're going to hit the high density and the low density areas. The mistake is to aim, as Koustubh kind of indicated in his part of the talk, if you don't do some sort of randomized design in where you put your traps, the tendency is to put them in the higher density areas, and then you will get very misleading abundance estimates. So you don't have to cover a large fraction of the area, but you do have to cover it in a statistically rigorous way.
Thanks, David. That's very, thank you. That helps clarify some. And I want to ask Koustubh if he has anything to add to that.
No, I mean, that's exactly what I tried to briefly talk about towards the end of my talk, which is where you sample is really crucial, right? And if you end up biasing where you're sampling just because, oh, I can see a lot of snow leopards there, I'll get a lot of camera trap pictures on over here, then you are quite likely to bias your results. By the way, Justine, if you're okay, there's a very interesting question, and I think, David, you might be the best person to help answer this. This is by Becky May. She's asking that PAWS is working towards establishing more robust baselines for snow leopard estimates. What's your view and advice on how to set up monitoring systems, which mean we will be set up to compare estimates over time? Yeah, how can we have another estimate which is comparable to indicate population trends? Is this embedded in the guidance, or how are we looking at it?
Yeah, I believe, well, you can answer about the PAWS guidance. At the moment, as I understand it, PAWS is looking for the baseline, right? So we haven't begun to address the issue of how you estimate population trajectories. But that example I showed from Baylis is a good example for that. So to get a decent estimate of population trajectories and demographic parameters, you really need a time series of estimates that is around at least as long as the animals live. So for snow leopards, I'm guessing you want a time series of estimates of around 10 years or so before you're going to be able to tie down demographic parameters reliably. But the demographic parameters, I once wrote a book on closed population abundance estimation, and it went to a reviewer, and the reviewer was confused because he said, who is interested in closed populations? No one wants to know about closed populations. What we're interested in is trajectories. So I think the ultimate end game must be understanding the demographics and the population trajectories. But you need a decade or so of randomized, well-designed surveys in order to be able to do that reliably.
Thank you, David. I think that truly answers the question well enough.
Thanks. And we have another one here, another question from Hamera that was a bit before, and she's wondering about how models can be comparable based on your experience. So which of the existing models would you serve as more useful fit to get comparable population data based on your experiences? So maybe, yeah, go for it, Dave.
Well, so I don't really understand the question. So I've just described very, very briefly the absolute basics of spatial capture-recapture, but within spatial capture-recapture, there's really a huge variety of kinds of models. So you can put covariates into the encounter rate function, you can put in the density function, you can make individuals respond to traps or not respond to traps, and so on and so on and so on. And I'm not sure if I'm answering the question, but there are sort of standard statistical model selection tools where you can investigate models. For example, does density depend on ruggedness? Does it depend on estimates of prey or so on? And you have objective ways of choosing between those models. Is that answering the question or not?
Let's see if she writes it in the chat, Hamera. Please feel free, Hamera, to add on if it doesn't answer your question. But then maybe I just to jump in and ask David, what is the one recommendation then that you would have for national teams that are actively carrying out camera trap surveys for snow leopards and plan to use spatial capture-recapture models, what is one recommendation you would tell them right now? Because I think there are many range countries here.
The single most important thing I would say, and the single most frequent failing, is not defining your survey region in advance and not designing your survey with some randomized, statistically rigorously. So the tendency is to put the cameras out, and after you put them out, decide where you want to draw inferences. You can't do that. What you need to do is decide what population, what area you want to estimate in, and then use some sort of stratified or randomized design within that area. So I would say design is the most important thing and the least well done thing consistently.
Absolutely. I'll second that. I mean, having gone through several years of working with David and seeing the statisticians' levels of frustrations vary at very different things, design, I think, is the most important issue. It really can make or break a survey. And often that's what we give the least emphasis on. Most of the time, just go out, set up the cameras, and we'll decide everything else later. And that's where you end up with the challenges that you're just scratching your head with. How do you deal with it? So I'll second that entirely.
And adding to that, Hamera's come back asking you, so, okay, she agrees with potentially with designs, but she's asking about what about some areas that are very rugged, more remote, with higher barren mountains? What would, with few sympatric herbivores, what kind of approach would you suggest for these kind of, I mean, in terms of design, what kind of design is most applicable for areas where you can't all access? If you can give some, I don't know, an example perhaps might help.
So again, I'm not 100% sure I understand the question. And in answering the question, the closest I have to say the closest I've been to a snow leopard is seeing a picture on a computer. So in terms of inaccessible areas, and that, I don't really understand.
But you've been to Bishkek, which is 35 kilometers from the nearest snow leopard. So you've been that close.
That's true, but all I've seen is the mountains in the distance, not the actual snow leopard. So I do appreciate that they live in very inaccessible habitats, and sometimes where the statistician tells you to go, you just can't go. And maybe that's what this question is about. So if that is the case, then I guess the advice is, in order to be able to estimate in all types of habitat, you've got to, if you think of the habitat as a covariate on your x-axis, so here's good habitat, here's bad habitat. If you only put your traps down here, and then you try and extrapolate what's going up here, you're going to get a bad estimate. It's very dangerous to do that. You've got to put your traps in a way that samples across the range of your types of habitat as far as you can. And then you can use a model to predict into the bits in between that you didn't sample. I don't know if that answers the question well.
Thank you, David. And maybe we can take you up on your suggestion right now before we run out of time. Do you mind sharing that extra part about where SCR is going and the future of SCR?
Yeah, sure. So just give me a second. Share screen. Okay, so that should be my screen saying some extensions. Is that what you see?
Yes.
Okay, so I'm just going to talk about some developing areas in spatial capture-recapture. And given the questions, the one thing that I really haven't included here, but should have, is how you deal with uncertain recapture identification. So that's very much a hot topic. But I haven't prepared anything on that. So let me just go through the stuff that I have done. So typically in capture-recapture, you're used to thinking of capture occasions. Now that made sense when there were traps and you were catching animals and then you had to release them. But with a camera trap, it doesn't really make sense. The camera trap's out there all the time. It just surveys continuously. So really, we can look at models that don't have occasions and have continuous time in. And when you do that, you can get some really useful inferences about animal behavior, for example. So this, again, to illustrate this, is the study of jaguars in Belize. It's just in an area in Belize, and these dots are the camera traps. And the picture over here is showing, this is the survey region, the sort of grayish region. The black dot is a particular animal's activity center. And then we've used this spatial covariates here in order to fit an encounter model that isn't a symmetric normal bump. It depends on the habitat. And in this case, the habitat is distance from river. There's a series of rivers through here. So you can see that this animal, there's actually a river running sort of east-west through this region here, which is why the activity, the range, the green area, which is where it spends, the animal spends more time, extends further to the right than it does up and down, north and south. That's not the most interesting part of this. The most interesting part of this is that you put continuous time in, and I'm going to now do an animation that runs through a 24-hour cycle, and you can see how you can draw inferences about how the animal's level of activity and range of activity changes with time. So let me just run through that cycle. So here we go. This is midday, it contracts. The animals are crepuscular. Now we're getting towards evening, midnight, early in the morning, midday contracts again. So not only do you get inference about where the animal goes in relation to the habitat, but you also then get inference about how where it goes changes with time. So that was one extension, putting continuous time in to look at activity patterns across time. The other one to talk very briefly about is GPS collar data. So someone did mention this, saying, well, you know, you get a lot of information about GPS collars about movement. And so that surely must tell you about the encounter functions that I spoke about that you estimate from camera traps. And the answer is yes. It provides a huge amount of extra information on how the animals use their habitat. So these dots here are from, I think, 12 snow leopards in Tost, with each color being a different animal. And so you can see, if you remember their habitats, this was high ruggedness down here where there are lots of dots, and then they basically follow the ruggedness here. Now you can think of this GPS data as pretty much like spatial capture-recapture data. The spatial capture-recapture data, you got counts, number of times the animal was seen at the traps. What this data is number of times the animal was seen at each grid cell. So each little dot here is a pixel. So it's sort of like a camera trap study in which every grid cell is a camera. Not quite, but you can think of it that way. And so we can fit an encounter rate model to this. And we've done exactly that in this picture here. And the histogram is, along the x-axis is distance from activity center for all of the 12 animals. And the heights of the bars are number of GPS pings at that distance. Now, the reason that there are very few pings at zero distance is because at zero distance, there's only one point. And as you move out at greater distances, you get bigger and bigger circles. So there are more and more points. And therefore, as you move out, there are more and more pings until the frequency with which animals range beyond some distance starts the pings dropping off. So the point of this survey, this image really is just that we can fit those exact same encounter rate models that we fit to the SCR data, we can fit to GPS collar data, and much richer data source. You get many, many more observations. There are some caveats that go with this, and that is that the animal movement may not be exactly the same as from the ping data. GPS data may not be exactly the same as the detection data on cameras. For example, the animal may move and not encounter cameras. So you have to be a little bit careful about the interpretation of that. This slide I just wanted to put in response to Koustubh's comment. So what we did here was fitted a model in which animal movement depended on ruggedness, and then predicted where the animals would move. So I've just taken a few examples. An animal starts here, starts at the green dot, in order to get to the red dot would follow these lines. And you can see that the lines basically stick to the high ruggedness, and then it jumps across to another bit of high ruggedness and so on. So that's the sort of thing that Koustubh was talking about, the model, SCR model producing the sort of behavior that would be expected a priori. We can model movement directly. So with spatial capture-recapture, we think of there being a single activity center attached to an animal. But you can think instead of a whole path being attached to the animal, the path being where did the animal move. And we don't observe the whole path, just like we don't observe the activity center. So this is a path of an animal, the red, and this is a detector, and it was observed only at these 2 points, the green arrows, and they're not observed elsewhere. But the methods, very similar methods apply. And the nice thing about that is that because we're then directly modeling animal movement, we can then predict the time that the animal spent at any point in space, because you've got models for how it moves through space. So this is the Belize jaguar data again, and this is for a particular animal, contours of amount of time spent at any given point in space. The last thing I want to mention is modeling spatiotemporal correlation. Now, I'm mentioning this really because there's a cool animation that goes with it. But aside from the cool animation, the reason for wanting to worry about this is that all the capture-recapture models, spatial capture-recapture models, assume that where an animal is caught at on any particular occasion or any particular time is independent of where it's caught next. But that's not the case. And because animals, you know, if it's caught at a trap over here now, then the next time it's caught is likely to be in the vicinity of that trap, not in a trap on the other side of its home range. So this is applied to this Ngorongoro tiger data set. And those dots are the cameras, and I've just followed a single animal here. So this is a single animal whose activity center is at the red. And this was a long series, I think it was 60 or 90 days these cameras were out. And I've just split them into 2 halves. So the gray is the first half of this period and the red is the second. You can see that the location of the animal is very highly correlated with time. So in the first half, it tended to be in the south of its area. The second half, it tended to be in the north of its area. Now, we typically don't model that at all with spatial capture-recapture, but you can. And so this is a model that does that. And so these dots are the traps. There's the activity center, and the shading here is this normal bump, just shown as a heat map instead of a 3D bump. So that would be its default activity center and space usage. But I'll show an animation in which you get a star when it's caught on a trap, and then its space usage will shift in that direction because it's next likely to be caught in the vicinity. So let me just show that animation. Caught there, and then it's pulled in that direction. Caught there, pulled in that direction. Caught there, pulled in that direction, and so on. So you get this little higher likelihood of capture in the region that it's just been caught. And so you might say, well, that's a cool animation, but so what? Well, what is it actually can make a difference? Because this lack of dependence can lead to biased estimation. So we fitted with this data set, we fitted this model is with dependence, and then these are 2 other models without dependence. Point of this slide really is just that the model with dependence is selected by all model selection criteria as much better than the other models. Okay, does it make a difference to abundance estimation? And here's the really interesting thing about this is just look at the left picture. This is population size. So when we model the dependence, we get this red curve. This is a Bayesian inference. So this is the posterior distribution of population size. And this is just standard SCR, which assumed independent captures across time. And when we model the spatiotemporal correlation, we get much higher estimates. And in simulation, we showed that if there is spatiotemporal correlation, you can get quite negatively biased estimates if you don't take account of it. Anyway, as I said, the main reason for including that really was the cool animation. So that should give you a taste of some of the sort of developments that are going on and the sorts of things that spatial capture-recapture should be able to do in the near future.
Thanks, David. So we're running out of time, and I think that was really nice to hear about where SCR is going. But maybe Koustubh wants to finish with the last 2 thoughts on where PAWS is going. There have been a few comments on, are we thinking long-term? Will we be monitoring the species and not just having one estimate or one baseline? So I don't know if you have any last words you want to share with everyone, Koustubh?
Thank you, Justine, and thanks, David. That was a really exciting presentation about where we are headed to. So essentially, I'll just second what David said a few minutes ago, that nobody's interested in a single abundance, right? That's just telling us how many there are at a point in time. Ultimately, the entire purpose of this whole PAWS process is to be able to set a baseline, know what you have, and then monitor it in due course of time, so that we know how many snow leopards we are living with, and over time the hope is that at least the numbers don't decline further. So I think the long-term monitoring or long-term assessment of populations of snow leopards is really at the heart of it. So I think that's where it will head to. But just to add a couple of more points, that there's a lot of resources that are being generated and developed. There's a lot of publications which are coming out and will come out in due course of time. So we are really thrilled about how PAWS has not only facilitated this massive collaboration between maybe more than, let's say, 50 organizations and researchers and individuals, but it's also kind of bringing in a good scientific approach to on the ground. And I think that's to me an exciting development. And it's none of it could have been possible without the support of those who are doing it on the field and on the desks and in the labs. So really, it's an exciting development, and it's not as quick as one would have wanted it to be. It takes time to come up with the best models, the best methods, and an appropriate guidance. But again, we are grateful that we have this entire PAWS panel, which provides the right kind of advice and suggestions, whether it's about snow leopard ecology or population ecology. So I feel very empowered to get to work with with such a wonderful team, with such experience, that we will get there. We won't get into the valley of despair anytime soon. We've probably crossed over.
Thanks, Koustubh. And David, what about you? Any last thoughts before we leave? Because I'm just mindful of time. We've covered the hour, but do you have anything else you would like to share with the group, or any last thoughts about PAWS and how spatial capture-recapture is supporting PAWS?
Nothing really, except from my perspective, it's a really exciting initiative that's making a difference to the world. So I'm an academic statistician, and it's really nice to do something that has real impact and people think are important, and especially on such a large scale. It's kind of an unprecedented effort, and so it's really nice for me personally being involved in that.
And we are very grateful for that you are involved as well. I think it goes both ways. So I see, just before we end, I see there are many other questions in the chat. Feel free to write to us, to Reki or Koustubh, so we can follow up with some of these questions that were answered. I think there were specifically questions that were model related or related to SCR that we can follow up on if anyone's interested. But I wanted to just thank you both for coming here, and thank you also to the help desk for being there for snow leopard conservationists and practitioners across the range and across different countries, and just being there to answer questions that I just even simple questions that might sound obvious, as you've shown today, David. The obvious is not always that obvious. So thank you for being there to help our teams. And before we say goodbye, we just wanted to let you guys heads up for the next webinar. We're actually going to travel back in history and go back and discover one of the first PhD graduates on snow leopard conservation, which is Raghu, and he's going to share about his work in Ladakh on these initial days. So we look really forward to that in early August. But a huge thank you for David and Koustubh for taking the time to share with us about this very fascinating work and how science can come together with policy and really make a difference for snow leopards. So thank you both.
Thank you very much.
Thank you.
For the invitation.
Thank you.
Thanks to everyone. So I guess we can all log off. Thanks, and we stuck to time. Wow, David, Koustubh. That's the success in the webinar. Ciao.